Welcome everyone, and thank you for joining this session. I'm Rafael Martinez Arenas, an international exchange student from Spain. At the moment I'm studying here at Channam National University, and today, we'll delve into an exciting article innovating in the field of microbial electrochemical technology, hydrogen production in a single-chamber microbial electrolysis cell, or MEC, without a membrane. This research, conducted at Penn State University, explores a promising pathway for sustainable and cost-effective hydrogen production. Hydrogen is pivotal in the transition to renewable energy systems. Current methods like fermentation and water electrolysis, while effective, face limitations in efficiency and energy consumption. Microbial electrolysis cells, or MECs, harness bioelectrochemical principles to offer a more energy-efficient alternative for hydrogen production. Traditional MEC designs rely on membranes to separate chambers, but these add cost, complexity, and energy inefficiencies. The study we're discussing investigates the potential to eliminate the membrane entirely while maintaining high efficiency and hydrogen recovery. Let's talk about the scientific basis of MECs. At the core of this process is electrohydrogenesis, where exoelectrogenic microorganisms oxidize organic matter at the anode, releasing electrons and protons. These electrons travel through an external circuit to the cathode, where protons combine with them to form hydrogen gas. MECs operate under anaerobic conditions with an applied external voltage to drive the hydrogen production. The single-chamber, membrane-less design explored in this study simplifies the architecture, reduces costs, and minimizes energy losses compared to traditional dual-chamber MECs. The experimental MEC design was compact, using a 28 ml polycarbonate cylindrical chamber. The anode was a high-surface area ammonia-treated graphite brush, while the cathode was a platinum-coated carbon cloth. Sodium acetate served as the substrate, and the solution's conductivity was adjusted to 7.5 ms/cm and later increased to 20 ms/cm to assess performance under varied conditions. The gas production was monitored using respirometers, and hydrogen purity was analyzed with gas chromatography. The anode was enriched in a microbial fuel cell, or MFC, before transferring it to the MEC. The reactors were drained and exposed to air after each cycle to suppress methane-producing microorganisms. This innovative approach, coupled with nitrogen sparging to remove oxygen, ensured optimal anaerobic conditions for hydrogen production. The results were remarkable. Hydrogen production reached a maximum rate of 3.12 cubic meters per cubic meter of reactor volume per day at 0.8 volts with a conductivity of 7.5 ms/cm, increasing the conductivity to 20 ms/cm. Improved production rates for applied voltages between 0.3 and 0.6 volts, peaking at 2.61 cubic meters per cubic meter per day. However, high voltages and conductivity beyond these levels caused bacterial stress, reducing efficiency and production. Hydrogen recovery rates were equally impressive, ranging from 78% to 96% at applied voltages of 0.3 to 0.8 volts. Interestingly, methane production remained low at these higher voltages but spiked at 28% when the voltage dropped to 0.2 volts, due to prolonged reaction times. This demonstrates the importance of maintaining optimal reaction conditions to minimize contamination and maximize hydrogen purity. The energy efficiency of this MEC system was another key highlight. Electrical energy efficiency peaked at 406% at 0.3 volts, demonstrating the system's remarkable energy conversion capability. Substrate energy efficiency remained above 95% across all conditions, showcasing effective utilization of organic matter. However, higher applied voltages shifted the energy contribution away from the substrate toward electrical input. Compared to traditional designs, the membrane-less MEC simplified the system architecture and reduced costs without compromising efficiency. Hydrogen purity exceeded 87%, and overall energy recovery rates were comparable to or better than systems with membranes. These findings indicate that the absence of a membrane not only simplifies the design but also allows for higher current densities and production rates. This mech outperformed many traditional systems. For instance, a Nafion membrane-based mech achieved a recovery rate of 53% at a hydrogen production rate of 0.02 cubic meters per cubic meter per day. In comparison, the system under study reached a recovery rate of 86% and production rates over 1 cubic meter per day. This demonstrates the superiority of the membrane-less design in terms of both performance and cost efficiency. In summary, this study has shown that single-chamber, membrane-less mechs can achieve high hydrogen recovery and production rates, significantly reducing system complexity and costs. These findings have profound implications for the future of bioelectrochemical systems, making sustainable hydrogen production more accessible and cost-effective. The scalability of this technology could play a vital role in meeting global hydrogen demand.